Hi, micro folks. This is Professor Carberry, and we are continuing our series of micro lab demos for our Biology 440 course for fall 2020. This week we are exploring what our ancestors knew. This is the title in our Open Stack Microbiology book of what our ancestors observed dealing with microbial phenomenon. They didn't know the microbes existed, but they saw the consequences of microbial life around them. So we've been exploring my favorite topic, food, right? Using it as an example of what microbes can do. So again, folks, just want to show you from my refrigerator. We have a great example here of a microbial process. This is some lettuce that the microbes are starting to decompose, right? So with decomposition, food spoilage, generally we are repulsed by the smell, the consistency of rotted food, which is really good because often foodborne pathogens could be present or microbial toxins could be present. So this was a huge issue when food is limiting, and in the old days before refrigeration, depending on which part of the world you lived in, there would be a growing season when you could grow and then harvest your fruits, your vegetables, your grains, and then you had to find a way somehow to preserve the food so they wouldn't spoil, right, in the old days of refrigeration. So I just wanted, this is a little plum that fell off our plum tree, and I'm not sure if you can see this beautiful kind of cloudy opaque coating on the outside of the plum. This is going to contain lots and lots of microbes, right? And so some of those microbes potentially could cause food spoilage, as we saw with the lettuce, right? But some of these microbes potentially could carry out a process called fermentation that would actually help preserve the food by producing either alcohol, ethanol, which will inhibit the growth of spoilage microbes, or perhaps produce acids, which can also inhibit the growth of spoilage microbes. And again, folks, this is going back to our alcoholic fermentation. You'll remember that we were looking at some blueberries and grapes, and we cultured the microbes from the blueberries and grapes and put this is a TSA plate all-purpose medium that will permit both fungi and bacteria to grow. And you can see here the grapes the outside of the grape is chock full of microbes, and we see many different colony types telling us many different microbes are present. And this is the blueberry juice, right? And there's so many microbes present, all the colonies are growing together. And then, folks, this was the grape juice and blueberry juice after 1 day of fermentation. I know it's kind of hard to see here. This hasn't been 24 hours yet, but what I can see is there's been selection. Some of those microbes outcompeted the other microbes, right? And so there is, I don't know if we want to call it microbial warfare going on, but the microbes are going to be competing with one another for nutrients. And so what humans have learned kind of through serendipity, humans have learned how to select for the good microbes, right? And so with this, you guys, this is just the most incredible book by Sander Alex Katz. It's called The Art of Fermentation, with a foreword by Michael Pollan, right, who's a big name in food. This book, you guys, is phenomenal, but it gives us kind of a history of how humans have learned how to make food, change conditions of the food to encourage the growth of good microbes that will carry out fermentation and thus inhibit the growth of the spoilage microbes. And again, folks, historically this was so important because this permitted our ancestors to preserve food in the absence of refrigeration, right? And that was essential for survival. So I'll probably do another video just reading some of the fantastic information that Mr. Katz here has in this great book, The Art of Fermentation. And folks, just so you know, there might be a lot of background. This is garbage pickup day in our neighborhood, so we might hear the recycling trucks and garbage trucks going on. And it's also, thank heavens, a nice, breezy, cool day. We're not the 103 temperatures we've been having. Okay, folks, so we've already started some experiments looking at alcoholic fermentation performed by the yeast Saccharomyces cerevisiae. We have our blueberries, we have our grapes, hopefully undergoing the alcoholic fermentation by the wild yeasts, Saccharomyces, which is found in nature, just like on this little plum. We can actually try to make plum wine, right, just by mashing these little guys up and storing them under anaerobic conditions. So the fermentation we wanted to explore today is lactic acid fermentation. And lactic acid fermentation is performed by a group of bacteria called the lactic acid bacteria. And these bacteria include some pathogens like Streptococcus pyogenes that can cause strep throat, is one of these lactic acid bacteria. But it also includes lactobacillus, Lactococcus lactis. These are lactic acid bacteria that are beneficial. They don't cause harm. What they can do is take the sugars from plants or from milk and ferment the sugars into lactic acid. And lactic acid, again, you guys, as the name suggests, it's going to lower the pH. It's going to cause an increase in hydrogen ion concentration. And that low pH, that acid, will inhibit the growth of the spoilage microbes. And thus, we can preserve our food in the absence of refrigeration. So folks, I just wanted to give you an example. This is some sauerkraut, right? Sour cabbage that I made about 6 months ago, right? And I kept it in jars and then took it out. And I'm going to just take a little taste here, you guys. And compared to fresh cabbage, wow, this is sour, right? And that sour taste is telling me that there's lots of hydrogen ions present. It's a low pH. Our taste receptors, when they're interacting with chemicals, with food, if there's really low pH, high hydrogen ion concentration, we sense that as sour, and thus sauerkraut, sour cabbage, right? What's the advantage? That low pH is going to preserve the cabbage in the absence of refrigeration, so we don't get the spoilage like we saw with the lettuce. Okay, so folks, when I was growing up, my father taught me how to make sauerkraut, and so this is just a little demo. So we would use clean mason jars, and we would put a layer of chopped cabbage, and this is kind of an abbreviated demo, you guys, on this. And then we would sprinkle salt. We put a layer of cabbage, and then we'd sprinkle salt on it, and then we would pound it down. And then we would do this sequentially, right? And we keep pounding, and it was really important that we got the water, the cabbage juice, to flow out of the plant cells. And now I understand what we were trying to do is create an anaerobic environment, meaning no air present, no oxygen present. And by creating that anaerobic environment, you guys, we're going to favor the growth of those lactic acid bacteria, or if there's Saccharomyces present, we could help favor the growth of the Saccharomyces. Now, my thought is, by adding the salt too can inhibit the growth of spoilage microbes. And I am indeed wondering, you guys, if the salt maybe inhibits Saccharomyces, so we don't get alcoholic fermentation, and it permits for the growth of the lactic acid bacteria. And again, I'm always trying to find connections here, guys. We have good lactic acid bacteria on our skin, on our mucous membranes. And my thought is, and I'll have to check this, is maybe because some of these good lactic acid bacteria maybe have evolved mucous membranes, junction between mucous membranes and skin. Maybe they're a little bit more salt tolerant. I'm going to have to check this out. We can do an experiment to see if the lactic acid bacteria are more salt tolerant. Okay, so that's one way to make sauerkraut. So I've been really interested in this. So yesterday I was going to do this experiment. I had an old bag of chopped up cabbage. Right, so you can get almost a pound of chopped cabbage at grocery outlet for 99 cents, right? And the bag of cabbage I had was, it was about almost 2 weeks beyond the use by date. And so when I opened it up, I could already smell this wonderful aroma of lactic acid. So I was like, oh, the lactic acid bacteria, they beat me to the punch, even before we'd done anything, right? So this to me, you guys, is so interesting. Like some of our folk traditions, like how we prepared, say, the sauerkraut, like which parts were essential, which parts are essential to get the good lactic acid bacteria grow, and which parts of the process maybe we don't need. So what I thought we could do is run our own experiment, and we'll call this the cabbage and lactic acid fermentation experiment. And what I thought we could do, you guys, is we could use two variables. So one would be the presence of air, so an aerobic, and the opposite variable would be an anaerobic. Anaerobic growing conditions. And then the next one, you guys, would be, we could remember how my dad taught me to use salt. I thought, well, let's see if that salt is important. So we'll also introduce the variables of no salt or salt and see if the salt actually does encourage the growth of those lactic acid bacteria by inhibiting the growth of others. And then, folks, another thing I thought I would do, all of this will be run at room temperature or garage temperature. But then I thought we would have another, like a negative control, and that would be just the cabbage in its bag, just put back in the refrigerator so we can see maybe the impact of temperature. And then one last thing, you guys, when I was in college, I had a wonderful opportunity to go to school at the University of Edinburgh in Scotland. And during the winter breaks and the spring breaks, I would go up into the Highlands to work on the farms there. And so during the winter break, I worked on a dairy farm, and during spring break, I worked on a sheep farm. And in Scotland, since it tends to be kind of rainy, instead of making hay, which is dried grass to feed to their cattle, they make silage. And basically, you guys, silage is when we're going to preserve the nutrients in the grass, prevent them from spoiling by encouraging lactic acid fermentation. And there's an art to making silage, you guys, just like there's an art to making these fermented vegetables. And one of the experiments our microbiology class did was to kind of make our own silage, changing the conditions. And one of the conditions was to add excess water. So you guys, just for fun, I'm going to add one of our experiments will be the chopped cabbage in excess water, aerobic, no salt. Okay, so that's my experimental design. So what I did, folks, was, again, I just bought the commercially prepared chopped cabbage. This is green cabbage, purple cabbage, and some carrots. And I just used plastic bags, and I measured into each bag about 2.5 cups of cabbage, and that was about 7 ounces or 200 grams. So I wanted to make them all consistent. And then, folks, in the salt treatments, I'm going to add 1 teaspoon of salt, which to me is quite a bit of salt. My daughter just told me you're only supposed to have 1 teaspoon of salt a day, for a healthy diet. So 1 teaspoon of salt per bag is going to be a lot. So then, folks, we'll have aerobic no salt, we'll have anaerobic no salt, we'll have aerobic salt, and anaerobic salt. So again, folks, because I know these videos get really long, so what I did was I aliquoted the bags of cabbage. And so I've just written on the bags in this case aerobic and salt, and then in this bag here's just anaerobic and salt, and then the other bags, folks, we're not going to add any salt to. And so we have aerobic no salt, anaerobic no salt, and then we have, and let me just grab this one if I can, and then one of them, you guys, I just added excess water, and I didn't use tap water because it has chlorine in it, right? So I want to see what will be the impact of having that excess water. So all we're going to do is we're just going to add a teaspoon of salt to our 2 salt treated, guys. Okay, so about a teaspoon of salt, which is quite a bit of salt. And then, folks, I'm going to mash these up a little bit, because remember how we said in the traditional production of sauerkraut, and I don't know about kimchi, folks, if in kimchi you pack your vegetables in, like, we were kind of pounding the cabbage. So if anybody is a good kimchi chef, let us know. That would, gosh, wouldn't that be great, you guys, if we were all together and we could just have like a fermented vegetable party. Okay. And then, folks, just what I'll do is I'm going to I'll seal these, and then I'm going to massage the cabbage. I'm going to try not to do it because the crackling of the bags is really irritating. But I'm going to try to work the cabbage and carrots, kind of getting them to break down, releasing some of that fluid. And then with the aerobic ones, I'm going to try to not seal the bags tightly, right? So air will have access. And then the anaerobic ones, you guys, again, I'm going to massage the cabbage, trying to get the plant juices released. And then I'm going to try to roll it to get rid of any excess oxygen. Seal it, well, actually, roll it to try to get rid of the excess oxygen, the excess air. Seal it, and then what I'm going to do is stick them into a mason jar. Now, normally, you guys, fermentations are best done in glass, and I feel really bad that I'm using these plastic bags, but I will try to reuse the plastic bags. Okay, so what I would do is I would try to squeeze out as much air as possible, seal it, and then I'm gonna store it, you guys, in the mason jars that have a little rubber gasket there. So this hopefully will help us try to keep it as anaerobic as possible. Okay. And then, folks, again, I won't show the techniques because we saw it in the previous alcoholic fermentation. I'm also going to plate some of the microbes from the juice onto TSA and Sabrod's dextrose medium, and I'll also take some samples to make a smear so we can gram stain them, so we can see. Hopefully what we can see is a sequence of who is present on day 1, who's present on day 2, who's present on day 3, and maybe we can see kind of this microbial competition going on and see who is selected using these different conditions of presence or absence of air, presence or absence of salt, and again the excess water. Okay, folks, I think that's it. So later today I wanted to let our grape and blueberry fermentation just finish until this afternoon. So then I think that's going to be a total of 48 hours. And it's really cool because already just looking at the plates, I can see there's selection going on for some specific types of microbes. So I think that's a pretty cool experiment. Okay, you guys, let's see if we can finish this up. Oh, and then just as I'm closing up here, you guys, on another video, we're going to do another type of lactic acid fermentation using milk, which is another classic way to make fermented dairy products. That's a great way to preserve the nutritive value of milk in the absence of refrigeration. Okay. Oh dear. Uh-oh. I hope maybe this didn't work. Oh no. That would make me so sad.